Hey guys, I am here to discuss about yeast artificial chromosome in this video. So these are the contents. Let's start with the basic question. What is yeast artificial chromosome? It is actually a plasmid shuttle vector capable of replicating and being selected in common bacterial hosts such as E. coli as well as in the budding yeast Saccharomyces cerevisiae. The development of YACs were based on the logic that a eukaryotic linear chromosome needs for its replication and stability. Not only replication origin, but also the centromere and the telomeres are equally essential for an efficient replication and stability. So these are the basic components of yeast artificial chromosome, centromere, which help in efficient segregation of the chromosome into the daughter cells. Here present the centromere. CN4 here represent the centromere of chromosome 4. Then comes the telomeres. Here present two telomeres which preserve the integrity of the ends of the linear chromosomes. Then autonomous replication or replicating origin or ARS, element required for replication and preservation of YAC in yeast cells. This is here present ARS. Then col E1. The col E1 actually also act as an origin of replication, which useful for amplification of the vector in E. coli. Here is the fine structure of a YAC vector, which is 11.4 kilobase pair approximately. Here presents a two telomere, which is two ends of which contains restriction sites for BAM H1, and this segment also contains histidine 3-gene, and the segment also known as Stafford segment, which is the non-essential part of the vector, which can be cut down without affecting the vector. Here present uracil 3 and tryptophan 1 act as marker for selection of transformants, and here present the SNAV1 or EcoR1 restriction sites, which contains suppressor 4 gene. Here it is also a Stafford fragment, and here contains centromere of chromosome 4 and origin of replication. So restriction endonucleoside SNAV1 that produces blunt ends. SNAV1 site is used as the cloning site and is located within the suppressor 4 gene. Actually, those telomeres present here are from the protozoan tetrahymena, which have been found to be functional in yeast. So to construct a yeast artificial chromosome, we need to follow the 6 steps. Let's discuss it about elaborately here. Here is a circular YAC vector which has the potential to replicate inside E. coli, but it can't amplify inside yeast, because to amplify inside yeast it needs to be linearized, and to do that we need to treat this vector with BAM H1, which can cut this vector at this side and this side. So it will make a linear fragment, a linear segment. It's shown here, and here the EcoR1 site is our real insertion site. So to open that insertion site, we need to digest with EcoR1, which cut down those segments and open up for insertion. To insert our gene of interest, we also need to digest our gene of interest with the same restriction enzyme, that is EcoR1, to complete our insertion. And after that we ligate through different ligating enzyme, and finally the YAC has been formed, which is ready to transform. So yeast artificial chromosomes are special linear DNA vectors that resembles normal yeast chromosome. YACs contain telomeres that stabilizes chromosome ends, centromere that ensures chromosome partitioning between two daughter cells, and a selective marker gene which help in selection of transformants. YAC factor can carry hundreds of thousands of base pairs of foreign DNA, basically up to 2 megabase pair. When the YAC factors transfected into the yeast cells having mutations in the tryptophan 1, uracil 3 and adenosine loci, some yeast cells get transformed while others are not. To select the transformants from the non-transformants, we need to put them in a medium lacking tryptophan and uracil and containing limiting amount of adenine. The non-transformants will not able to survive because they are lacking tryptophan 1 and uracil 3 gene, but the transformants will survive. Generally the colony looks like red, but after a few hours those yeast cells having no foreign DNA will become colorless, but those yeast cells having foreign DNA causes the insertional inactivations of the suppressor 4 gene, restoring the red color. The reason behind the change in color is due to the activity of suppressor 4 gene. Suppressor 4 gene product is a tRNA that suppresses the mutations in the adenosine gene of yeast, resulting in a change in the color of colonies from red to white in the presence of limiting amounts of adenine in the medium. Here are some uses of yeast artificial chromosomes, including generating whole DNA libraries of the genomes of higher organisms, and the study of regulation of gene expression by cis-acting controlling DNA elements, to identify essential mammalian chromosomal sequence necessary for the future construction of specialized mammalian artificial chromosome or MACs. Thanks a lot guys for watching this video, and for such more videos, please subscribe my channel Knowledge Hunter.